So today we are going to study the techniques which are being used to study the chromosomes. So as you know, the chromosomes have centromere, then it has the small arm which is known as the P arm, then it has the Q arm which is the long arm. So this is the Q arm, this is the P arm, this is the centromere, and at the end you have the telomeres. These are the telomeres. And you also know there are 22 pairs of autosomes and one pair of sex chromosomes. But as you know, in many diseases, for example, in case of the AML, you will frequently find the translocation in between 8 to 21 chromosomes. Similarly, in case of the CML, you will find the translocation in between 9 and 22 chromosomes. It means some part of the chromosome is translocated to the other chromosome, and therefore the other chromosome become large and the one chromosome will become smaller in size. So how you can study these translocations or deletions or amplification, how you are going to study that? For that, uh, these are the things which we are going to study in this lecture. So AML is basically the leukemia. Both AML and CML is the leukemia, acute and chronic myeloid leukemia. Now, the one technique which is a very old technique and that is used to study the chromosome is the banding technique. So in the banding technique, you will see the chromosomes. Chromosomes have bands. Okay. So this is a staining technique which is used to create a visible pattern of light and dark bands on the chromosomes. So how you can do this banding? For in this technique, basically the stain which is being used is the GIMSA stain, G stain. Okay, it basically stain the AT rich region. So what will happen? The AT rich region will become dark, and the GC rich region will become um, lighter. So you will see the band pattern, dark and the light bands on each chromosome, and each chromosome has its own banding pattern. And on the basis of that, you can find it out any structural modification in the chromosomes. When we are saying the structural modification, it means uh, when we are saying the structural modification, it means you are going to study if there is any translocation, inversion, deletion, like this you can study. So what will happen in this banding pattern? As I said, the regions which are AT rich regions, they will become dark and the GC rich region will become lighter. So in this technique, basically two kind of the dyes which are being used is the GIMSA dye. GIMSA dye has three components. One is the azure dye, which is a basic dye. And when we are saying the basic dye, it means they are the cationic dye. They have the positive charge. And because they have the positive charge, they will bind to the DNA. Because DNA has the phosphate group and phosphate group is negative charge. So the DNA will bind to this azure dye because of this negative and the positive charge. Now the another dye is the eosin bi dye, which is the acidic dye, means this is the anionic dye, and it will bind to the positively charged proteins in the cytoplasm. The third one is the methylene blue. Again, this is the basic dye. Again, this is the cationic dye, and that will stain the acidic components like the nucleus. So when we are talking about the gene size staining, when you have the chromosomes and you are staining these chromosomes with the gene size stain, what will happen first? Like this. You can see this is the azure dye. Because this is positively charged, that will bind to DNA, which is negatively charged. Okay. Now eosin Y, because eosin Y is again the negatively charged dye, because this is the anionic dye. This dye will bind to this complex azure and DNA. Now this whole thing is forming a complex eosin by azure dye and the DNA dye. So, sorry, and the DNA. Then you are talking about the methylene blue dye. So methylene blue dye, what it will do, because it is again the positively charged dye, it will try to bind to the DNA, and so it will compete. Methylene blue dye, there will be the competition between azure dye and the methylene blue dye. And because of this, what will happen? You will be having a good contrast, and you can see the bending pattern in a better way. So this is the whole principle of the bending in the, if you want to study the chromosomes or structural modification in the chromosomes by the bending pattern. So you will get, in this case, you will get purple or a sort of violet color chromosomes you will get. Why we are adding the methylene blue dye? So there is not oversaturation of this color, the the color that is being produced by the azure dye, and because of this you will be having a, when you are adding the methylene blue dye, it will compete with the azure dye for the DNA, and because of this diffusion, the color brightness or the color contrast will increase, and you will get the, you will see the chromosomes either in the purple form or in the violet form. Now the another thing is reverse staining. In the gene size staining, in the G staining, you are staining the AT rich region. So what will happen in the case of G staining, if we talk about, in the case of G staining, you will get the AT rich region, darker band, GC rich region, light. In the case of reverse staining, this is reverse. You will get GC rich region, dark, and AT rich region, light. And why it is so? Because in the case of reverse staining, you are denaturing the chromosome. And because the AT has only two double bonds. In between A and T, there is two double bonds. In between G and C, there is three hydrogen bonds. So when you are denaturing the chromosome, this will denature first, and it will leave the GC rich region intact. And these GC rich regions you can study in the case of reverse staining. So when you are staining, when you are doing the staining of chromosome by the gene size staining, you will get something like this, the purple or violet color chromosomes you can see. In this you cannot find it out the the homologous pair of chromosomes, which one is coming from mother or which one is coming from father. So all the chromosomes are scattered, and they have the certain sort of bending pattern. Also the centromere position, according to the centromere position also you can recognize whether this is chromosome number 1 or chromosome number 2. Similarly, on the basis of length also. So if you want to study them, you can see it by the length, because sex chromosomes are smaller. So on the basis of length you can recognize them. On the position, on the basis of the position of centromere, If it is in the center, metacentric. It is if it is near to the end, acrocentric. Submetacentric means near to the middle position. This is submetacentric. So every chromosome has a different length as well as a different bending pattern as well as the different centromere position. And on the basis of that you can recognize them. Now coming to the second method, which is a karyotype. So what happened in the case of karyotyping? Karyotyping, you can see, karyotyping is usually done in the metaphasic state. So what you have to do, you have to stop the cell division. Whenever the metaphase is converting into anaphase, you need to stop it, and you can do it by the Colchikin type. This will prevent the transition of the metaphase to the anaphase, and because of that, all the chromosomes will be in the metaphasic state. And why in the metaphasic state? Because the chromosomes are very much condensed in the metaphasic state. So in the case of karyotyping, you can see in the gene size staining, the chromosomes were scattered. They were not in the pair. But in the case of karyotyping, you can see the chromosomes are arranged in the pairs, and that we have to do it manually by using certain softwares. The software will find the chromosomes on the basis of their pattern and length and centromere position, and it will arrange all the homologous chromosomes very near to each other. For example, this is chromosome number 1. These are the homologous chromosomes. One is coming from mother, one is coming from father. Similarly, like this. Now you can see the 8 and 21 translocation. This is the chromosome number 8. This is the chromosome number 21. You can see the chromosome number, the part of the chromosome number 8 is translocated to the chromosome number 21. And because of that, what happened? The chromosome number 8 becomes smaller and chromosome number 21 becomes larger. And if this is the condition, then this is leading to the acute myeloid leukemia, which is the blood cancer. So that in the karyotyping, the chromosomes are arranged in the pair. If there is a trisomy 21, so you will get the extra chromosome here. In case of the trisomy, in in case of the trisomy, you will get the extra chromosome here. Okay, so you can say this is the trisomy then. Trisomy 21. So what is the karyotyping? This is a visual representation of the all chromosome in a cell arranged by the size and the bending patterns. Detects numerical changes, means if there is trisomy, monosomy, it can detect. If there is any structural abnormalities like translocation, as I showed you in between 8 and 21, if it is a translocation, then this is leading to acute myeloid leukemia. So in this, the chromosomes are stained. The dye is Giemsa dye only and arranged in pairs under a microscope. A standard karyotype shows all 46 chromosomes in order. You can detect the numerical changes, structural abnormalities, but you cannot detect the mutation. Like if you want to see is there any mutation in the P53, you cannot detect it by the karyotyping. Okay. The other method is fluorescence in situ hybridization. In this you can see this is the chromosome, and let's say if we want to study this particular region, and we design the probe. The probe could be directly labeled probe, means they are labeled with the fluorescence molecule directly, or they are indirectly labeled, means this is the probe and it is labeled with the haptin molecule, which is not giving any kind of fluorescence. So what will happen? But this is, if you are using the direct probe, what will happen? Direct probe will bind to the target site. Once it is binding to its complementary site, then it will give the signal, fluorescence signal. But if you are using the indirect probe, then you are using the hapten molecule. Suppose in this case, let's say hapten, if you are using biotin as a hapten, then there is one extra step. You need to add avidin and HRP. So avidin will bind to biotin and HRP is a horse-radish peroxidase enzyme. So this enzyme, what it will do, you need to add the DAP, which is the diamino vanillate, and it will produce the brown color product. Okay. So if you are using the indirect probe, if you are using the indirect probe, you need to add the extra step. Suppose if you are using the biotin, then you need to add the avidin and HRP. You also need to add the substrate, so it will give the, if there is a binding, it will give a brown color product. If there is no binding, you will not get the brown color product. So like that. So this is the mechanism of fluorescence in situ hybridization. So the basic elements are the DNA probe and the target sequence. This is the DNA probe. This is the target sequence which you want to study. Before hybridization, the probe is labeled either indirectly or directly. Okay. So this is the indirect probe. This is the direct probe. The labeled probe and the target DNA are denatured to yield single-stranded DNA. They are then combined, means there is a hybridization, which allows the annealing of complementary DNA sequences. And if the probe is labeled indirectly, an extra step is required for visualization of the non-fluorescent hapten. So what, like I said, if there is a biotin, then you have to add the avidin and the HRP that uses an enzymatic or immunological detection. This hapten could be like deoxygenin also. If this is deoxygenin, then you need to use the antibody against deoxygenin, and that antibody should be fluorescence labeled. Finally, the signals are evaluated by the fluorescence microscopy. So this is the principle of fluorescence in situ hybridization. So there are many kind of probes which can be used, like the whole chromosome painting probes. Like you think about it, you got a noble translocation in a patient. Like let's say X to 10 translocation. Noble translocation you got. Okay, this is the X to 10 translocation you got. You have the probe against the 10 but not against the X. Then how you need to design the probe, and for that you need to design the probe against the whole X chromosome, and this is the whole chromosome painting probes. In these probes are going to label the full chromosome, but this technique is very costly. The whole chromosome, I would say 1 lakh rupees in India, or maybe $10,000 something in America also. The whole chromosome painting probes are complex DNA probes derived from a single type of chromosome that has been PCR amplified and labeled to generate a paint which will highlight the entire chromosome. Okay. Now coming to the repetitive sequences probe. These, the telomere has the repetitive sequences. So if you want to study the telomeres, you can use the repetitive sequences probes. These probes are basically bind to the repetitive sequences. Locus specific probe, if you know at this particular locus you want to study the mutation and you are very sure this should be the mutation at this particular locus, then you can use the locus specific probes. And these probes can be generated by using the cloning vectors like plasmid, bacteriophage-derived artificial chromosomes, yeast artificial chromosomes, depending on the size of probes. If it is like 150 to 350 kb, you can use yeast artificial chromosomes like that. Then this we are talking about the how many kind of probes we can use. In these kind of probes, the very much the good probe which can give you the good information is the whole chromosome printing probe. Then coming to the centromere fish. This is the fluorescence in situ hybridization method which is being used to study the infertility in the males. So what this will do in this particular fish, the probes which are going to be used, they labeled, they are the probes for the centromere position. They are the probes for the 1q. 12q means the larger arm of the chromosome, and then the 1p36.3 satellite of the chromosome 1 means the smaller arm. For the specific detection of the duplications and deletions of the 1p turn and the 1sen for the identification of the chromosomal breaks within the 1sen 1q12 region in the human sperm. So when we are saying the 1p turn means 1 is the chromosome number 1, p is the small arm of chromosome, turn is the terminal position. When we are saying the 1 centromere and 1q means it means, let me just show you. You can see this is the centromere, this is the smaller arm, this is the larger arm, and there is the region which is 1q12 region, which is very much near to the centromere. But if there is any break, then the binding will not be proper. You will not get the good signal in this. And in this way you can find it out that that this could be the reason of the infertility. So this is the centromere, this is the P arm, and this is the Q arm. And in the Q arm, this is the particular region 1q12 in the chromosome number 1. If there is any break in between centromere and 1q12, you will not get the proper signal, and by this you can identify the infertility in the males. Then coming to the cytochalasin B. Cytochalasin, what it will do, it basically stop the cytokinesis. So when the cytokinesis is stopped, you know that this is the status of cytokinesis, right? You have the two nuclei, right? So what happened? Why we are using the cytochalasin? So that we are sure that the cells are in the dividing phase. They are not in the non-dividing phase, because there are the non-dividing phase also, G1 phase, S phase, G2 phase. These are the non-dividing phase, and we want the cells in the dividing phase. Then we use basically cytochalasin, so we are sure that the cells are in the dividing state. This is the region this particular fish is basically used. So here you can see there are three chromosomes in one nucleus. It means there is a trisomy. Means there is a non-disjunction of the chromosomes. Means the chromosomes are not separated nicely. So in one nuclei there are three chromosomes, and in another nuclei there will be one chromosome. So this is the trisomy 21. And this technology you can basically use with the 24 color sky technology. Means 24 color sky technology means you can use the 24 colors to stain the 24 chromosomes. Okay, so you know the 22 pairs of the chromosomes, and then the sex chromosome which are 2, so there these are 24. So 24 colors you can basically use. Now coming to the LNA fish. This is a locked nucleic acid. So in this nucleic acid, what happened? You can see the bridge. There is a bridge in between the 2' oxygen and the 4' carbon. And because of this particular chemical modification of the ribose moiety, this LNA DNA probe, they bind to their complementary sequences with a very high stability. The stability means the TM basically is going to be increased. For example, uh, this is the RNA and DNA probe, and let's say we are going to study the mRNA, and these are binding to the RNA DNA probe is binding to the mRNA. The stability will be very high. The TM will basically increase. The melting temperature of this particular complex is going to be increased. So whenever you have the mRNA uh, and the stability is not stability is the concern of the fish, then you can basically use the LNA DNA probes, which actually increase the TM by 2 to 3 degrees. Okay, so if you want, if there is a hybridized complex, this LNA DNA and mRNA, and you want to break it, you need to increase the temperature at a you need to increase the temperature uh, like 2 to 3 degree higher than the natural probe, like that. Similarly, so these are known as the locked nucleic acid probes. And there is a structural shift from the B-type helix to the A-type helix, which has the higher thermal stability. This it will increase your melting. It will increase the melting temperature of the steel. Similarly, the same concern is with the peptide. They will also increase the melting temperature, more stable. This is the DNA, and you have modified it. You are making it a peptide nucleic acid, so you modify it or replace the phosphate bond, the phosphate bond with the backbone with the amide bond, okay, or the peptide backbone. This becomes a peptide nucleic acid. Again, the PNA DNA or PNA RNA probes, they are more stable because again they will increase the melting temperature very high. So like this. So whenever you know that the stability is the concern and probes are not binding with high stability with your particular target sequence, you can go ahead and use the PNA or the LNA probes. But there are very few companies which are making these probes, by the way. So quantitative fish, this is going to quantify number of telomere reduce. As you can see, the telomeres are at the end position. So if the telomere length is high, you will get the strong signal. If the telomere length is small, you will get the dull signal. So on the basis of that, there is a software which will measure this signal and will quantify the telomere length. This particular software is TF20, and it is important in studying the in role of telomeres in aging and cancer, because in aging, you know, the telomeres become smaller. Comparative genomic hybridization. So you are using the tumor DNA and the normal DNA in the equal amount, and you have the this microarray plate which has a thousand of probes which have the target sequences. Now this, suppose this particular probe is against P53, this particular probe is against MYC, this particular probe is against BRCA, something like that. So you are adding the tumor DNA and the normal DNA in the equal amount, and if suppose the tumor DNA is high expression of P53, then you will get the red signal. If it is similar, then you will get the yellow signal. If it is smaller, then you will get the green signal. So this is the comparative genomic hybridization to see whatever the sample you have has any particular sort of defect or not. So in this, the genomic DNA is isolated from both the tumor samples and the normal samples, and then you are going to label them with the fluorochrome and mixed in the presence of excess cord DNA to prevent binding of repetitive sequences. So you are not getting the background signal. Otherwise, the probes will bind to the repetitive sequences and you will get the background signal. In conventional chromosomal CGS, these are hybridized to normal metaphase chromosomes, and the ratio of intensities along each chromosome is analyzed. Why we are always doing everything in metaphase AGST? The reason is this, that the chromosomes are condensed. This is the region most of the techniques are being done in the metaphasic state. So increased DNA copy number in the tumor sample can be detected by the red fluorescence. Decreased copy number will be by the green fluorescence, and if it is equal, then you will see the yellow signal. So on the basis of that, you can identify. Suppose this is the P53 gene, then you can say the tumor sample has more P53. Okay, like that. P53 basically caused, apoptosis. So maybe you can see the green signal. So in the tumor sample, it is going to be less. So like that. You can see the another figure. This is the tumor DNA. This is the normal DNA. You label them with a different dye, psi 5 and psi 3. Combine them with in the equal amount. Then you are labeling them. Then red means this is the tumor DNA. Means this particular gene is very high in the tumor DNA. This particular gene is low in the tumor DNA. Like that. And this is the yellow color means this is equally in the normal and the tumor DNA. So this is explanation. DNA from the sample is labeled with the psi 5 and the psi 3. Then equal quantities are being mixed. Then they are hybridized to the array. Then digital imaging system are used to capture and quantify the relative fluorescence intensity of each of the fluorescence. The resulting ratio, on the basis of the resulting ratio of the intensity, that is proportional to the ratio of the copy number of DNA reference in the test and reference genome. If there is high tumoral DNA, you will get a red color signal. If there is a less, then you will get the green color signal. Okay. Like fish, there is a genomic in situ hybridization method also. So what it suppose this is particular plant, this is a triticale. Let's say this is the triticale chromosome. Triticale is wheat and rye combination, and you want to see in this particular species how many chromosomes are being similar to the rye or are coming from the rye. So what will you will do? You are going to label this rye with chromosomes with a direct or the indirect flow. After that, you are going to do the denaturation. Again, this chromosomes is also going to be, you are going to denature them. Then you are going to add the blocking DNA. Blocking DNA, what it will do, it will bind to the repetitive sequences. So what it will do, this blocking DNA will bind to the repetitive sequences. So again to prevent the background signal. So suppose this is the rye chromosome, this is the wheat chromosome, and these are the repetitive sequences. Then this probe will bind to these regions so that the background signal, the background noise is going to be less. Okay. Now, this is your target chromosome. This is the right chromosome, and the blocking DNA should be from the target species. So it is against the wheat only. You are adding this is in the high amount so that it will block the repeated sequences so that they will not bind to each other, and there will be less background signal. If you are not adding a blocking DNA, what will happen? These repetitive sequences will bind to each other, and then you will get lots of signal, unnecessary signal. After that, this there is a in situ hybridization. So this particular probe will bind to this chromosome. Blocking DNA will again bind to this particular chromosome. And then because this probe is labeled, so you will get the fluorescence signal. So again, I am repeating it. This is the target plant. This is the plant, like you can see. Okay. This is the right plant, the right chromosome. You label these chromosomes with a direct probe. Then you denature these chromosomes, and you are adding the blocking DNA also. You denature the blocking DNA. Both the blocking DNA and your probe will bind to this target chromosome, and the probe will bind to the very unique sequences, not on the repetitive sequences, because the repeated sequences are going to be blocked by this blocking DNA. So very unique sequences it will bind, and once it binds to the unique sequences, then it will give the fluorescence. So for example, you can see here, this is the wheat plant, and you are getting the green color signal that is coming from the rye DNA. So you can see this in the triticale, these many chromosomes are very unique, and they are coming from the rye DNA. So this is the technology which is known as the genome in situ hybridization. If you talk about the steps, you can see you are going to label the probe, you are going to blocking the DNA fragmentation, preparation of slide for your target chromosome, denaturation of the probe and the blocking DNA, addition of the probe and the blocking DNA in the hybridization mixture. Then you are going to denature the chromosomal DNA, and they will hybridize to the target chromosome. Detection of the chromosome probe in the chromosome of one parent, chromosomal DNA molecule of the second parent related to the unlabeled blocking DNA, visualization of the hybridization signal in the fluorescence microscope. Okay. Okay. So this is known as the GISH map. Okay. So that's all for today's lecture.